While it may seem a little odd that we're discussing the digestive system now, I hope you can use this information when considering the deliberation on whether or not you should be eating genetically modified foods. The digestive system's function is to break down the foods that we eat and to allow us to absorb them so that we can acquire nutrients and the building blocks that we need to make the biological molecules that we'll use to allow our body to function. There are a number of organs and accessory structures that are responsible for breaking down and absorbing foods, but we're going to focus on this general pathway. We'll talk about the mouth, the esophagus, the stomach, the small and the large intestine, and we'll briefly mention a couple of the accessory organs that facilitate digestion and absorption. Digestion is the mechanical and chemical breakdown of food, and we're going to be talking about both mechanical and chemical breakdown in all of the organs that were mentioned on the previous slide. Digestion begins in the mouth, and both mechanical and chemical digestion will occur there. Mechanical breakdown is going to be accomplished by chewing. Our teeth tear and grind the food into smaller parts that ultimately can be swallowed. Chemical breakdown occurs in the mouth as well, and it is accomplished by secretions through the salivary glands that are listed here. The components of saliva are shown here in this graphic. They include mucus and enzymes dissolved in a salt water that is listed as an electrolyte solution here. The enzymes that we're going to consider for digestion include amylase and lingual lipase. Lipase is responsible for initiating the digestion of fat in your mouth, and amylase is an enzyme that is responsible for digesting carbohydrates. This image shows starch as one of the major carbohydrates that we will digest, and that starch breakdown occurs via the action of the amylase enzyme in the mouth. Amylase will digest that complex carbohydrate starch into disaccharides, smaller components, and those disaccharides ultimately will be digested into monosaccharides, but that will occur in the small intestine. The chemical breakdown of food products the food that we eat will continue in the esophagus because the enzymes lipase and amylase will be mixed with our chewed food that we're going to swallow, and those enzymes will continue working in the esophagus. However, the esophagus itself doesn't contribute anything to the digestion process. It's simply a pathway from the mouth to the stomach. The chewed food, labeled here as a bolus, after it's swallowed, is going to be propelled toward the stomach via waves of contraction. Those waves of muscular contraction are called peristalsis. Peristalsis is going to be responsible for propelling our partially digested food throughout the digestive tract, not just in the esophagus. All of our digestive tract has a number of layers in it. Of course, the hollow tube that creates a digestive tract is labeled here as the lumen. The lumen is the hollow tube portion, or the hollow portion of the digestive tract tube. The inner layer of the digestive tract is called the mucosa, and it's the mucosa that's going to be producing the secretions that will facilitate sliding the food through the digestive tract, and the enzymes that are going to be responsible for breaking down our ingested food molecules. Just outside of that mucosal layer of the digestive tract is a connective tissue layer that contains blood vessels and nerves, and that's called the submucosal layer or the submucosa. Outside of the submucosa are layers of muscle that are responsible for peristalsis and also for mechanical digestion, as we'll see in just a bit. The connective tissue layer on the outside of the digestive tract is called the serosa, and it provides structure to the digestive tract. It says here that the muscularis layer, that muscle layer that is responsible for propelling the food through the digestive tract, it contains 2 layers of muscle tissue that is oriented in different directions. In fact, that's true, but in the stomach, there are actually 3 layers oriented in different ways that are going to be responsible for the mechanical breakdown of food that makes its way to the stomach, because those muscle layers will contract and they will sort of squeeze and move that food throughout the stomach, and that's mixing this, churning and mixing the ingested food, and that's contributing to the mechanical breakdown that initiated in the mouth. Chemical breakdown is also going to occur in the stomach. There are mucus secreting cells, enzyme secreting cells, and acid secreting cells in the mucosal layer of the stomach. Those things secreted by the mucosal layer are responsible for the chemical breakdown of food in the stomach. Hydrochloric acid is responsible for unfolding the 3-dimensional structures of protein. And once those proteins are unfolded, the enzyme pepsin, originally secreted as a precursor enzyme, pepsinogen, that is going to be responsible for breaking the chemical bonds that are holding amino acids together in proteins. So again, acid will unfold the proteins, and the enzyme pepsin will start breaking the bonds between individual amino acids to create smaller polypeptides in the stomach. This is an image of the small intestine showing the circular folds that increase the surface area of the small intestine. In addition to these folds, there are other structures that also increase the surface area in the small intestine. These include villi that are shown as the little finger-like projections in a micrographic image, as well as in a sort of cartoon image of what the small intestine surface might look like. Those villi, those finger-like projections in the small intestine, are created by cells growing out of these bases of the villi. At the base of each villus is what is called a crypt, and cells will grow out of these crypts to create these villi structures. An individual structure or finger projection would be called a villus, and many of them, the plural form of villus, are villi. So there are villi to increase the surface area in the small intestine, and on each villus are small microvilli that further increase the surface area in the small intestine. So while the image on the left shows these folds, what we don't see in that image are these finger-like projections called villi and the microvilli that line each of the villi. This is a table showing you all of the enzymes that are produced in the small intestine. They are responsible for carbohydrate digestion, protein digestion, and lipid digestion. While the digestion will be occurring in the small intestine, many of the enzymes that are responsible for this digestion are also created by the pancreas. The pancreas is one of those accessory organs that I spoke about earlier. The pancreas is responsible for producing several digestive enzymes that will be released via the exocrine portion of the pancreas into the small intestine to aid digestion. Another accessory organ, the liver, produces bile, which we'll talk about in just a minute. But that bile that is produced by the liver is stored in the gallbladder. And when food passes by the gallbladder in the small intestine, the gallbladder secretes that bile to facilitate digestion and absorptions of fats. While digestion that is initiated in the small intestine continues in the large intestine, the large intestine does not secrete any digestive enzymes on its own. So it's sort of like the esophagus in that way. Digestion that was started ahead of it in the pathway continues, but nothing really important with respect to our body occurs there. However, the bacteria in our gut are going to be contributing to some of the digestion. There are several different species of bacteria that live in our large intestine, and these are responsible for fermenting any undigested carbohydrates and proteins that make their way to that part of the digestive system. These bacterial fermentation processes are responsible for producing carbon dioxide, methane, and hydrogen sulfide, basically the components of our farts. In addition to completing, the digestive process, bacteria in our gut are also responsible for producing some necessary vitamins, vitamin K and vitamin B. If we take a lot of antibiotics because we have some sort of infection, we can kill off too many of these gut bacteria. And when we do that, the digestive process really doesn't occur the way that it's supposed to. So we can end up with diarrhea when we take a lot of antibiotics. So that's the digestive process. We break down food, but in order for us to get anything out of that process, we need to absorb the products of digestion so we can use them. The vast majority of molecular absorption is going to occur in the small intestine. These images show the increased surface area of the small intestine. And in fact, this is the function of that increased surface area. The increased surface area produced by the villi and the microvilli, as well as the folds that occur in the small intestine, allow for a greater amount of absorption in that part of our GI tract. Different things are absorbed in the small intestine. The first of those are monosaccharides. Remember, polysaccharide digestion or carbohydrate digestion was initiated in the mouth, and it continued in the small intestine, where those disaccharides that were produced by the action of amylase in the mouth are further broken down by enzymes in the small intestine into monosaccharides. Those monosaccharides are absorbed by the epithelial cells on the most inner layer of our mucosa. In addition to monosaccharides, we will absorb amino acids, the individual building blocks of proteins. Remember that protein digestion was initiated in the stomach, and in the stomach, proteins were degraded into smaller polypeptides. And in the small intestine, those polypeptides are enzymatically broken down to individual amino acids that are again absorbed by the epithelial cells of the mucosal layer of our GI tract. So we will absorb monosaccharides and amino acids in the small intestine, but we'll also absorb fatty acids. Absorption of fatty acids is shown in this image. Fat digestion begins in the mouth and is completed in the small intestine by enzymes that will break down those fats into individual fatty acids and monoglycerides. Those fatty acids and monoglycerides cannot be absorbed by the epithelial cells unless they are mixed with an emulsifier. Emulsifiers are found in bile salts. Again, the bile salts, bile was created by or produced by the liver, and those bile salts are stored in the gallbladder. When food passes by the gallbladder in the small intestine, the gallbladder releases the bile that it was storing. And in the small intestine, the bile salts are mixed with the fatty acids so that they can be absorbed by the epithelial cells on the inner layer of the mucosa. This table summarizes digestion, where digestion is initiated and completed both mechanically and chemically for all of the different food products. A few of the enzymes that we've spoken about I've listed on the right-hand side of this table. Finally, I hope that you've recognized that we have been talking about the digestion of biologic molecules. This image may look a little familiar. When we were talking about the creation of biologic molecules from individual building blocks, we saw that individual monomers form chemical bonds to create polymers. What we're talking about in digestion is the reverse process, where the polymers of the food products we eat are broken down into the individual building blocks, the monomers that make them up. We've spoken about the digestion of complex carbohydrates, polysaccharides, and starch into individual monosaccharides that will be absorbed. We've spoken about the breakdown of fats into individual fatty acids and monoglycerides that will be absorbed in the small intestine. And we've also spoken about the breakdown of proteins into individual amino acids that will also be absorbed in the small intestine. We have, however, neglected the breakdown of nucleic acids into individual nucleotides. And this is salient with respect to genetically modified foods. If we have modified the DNA in a food product, how are we going to digest that DNA? Well, in fact, DNA is going to follow a similar path to proteins. The chemical breakdown of nucleic acids will be initiated in the stomach because of the acid that is in the stomach. Those nucleic acids will be further digested by nucleases in the small intestine so that individual nucleotides, and in fact the parts of nucleotides, specifically the deoxyribose sugar and the ribose sugar of RNA, will be absorbed, as well as the individual bases.